स्टूडेंट्स, वेलकम टू बायोमिक्स सब। सो आज इस क्लास में हम डिस्कस करेंगे और एक पाथवे जो कि है पेंटोज फॉस्फेट पाथवे। ये जो पेंटोज फॉस्फेट पाथवे व्हाई इट इज रिक्वायर्ड, जो हमारा ग्लाइकोलाइसिस एक प्रोसेस था, इट वाज द ऑक्सीडेशन फेज, वन ऑफ द ऑक्सीडेशन फेज। द अदर ऑक्सीडेशन फेज वाज पेंटोज फॉस्फेट पाथवे। ये भी एक ऑक्सीडेशन पाथवे है। ओके, सो दिस पेंटोज फॉस्फेट पाथवे जो प्रेजेंट है उसको डिफरेंट डिफरेंट नाम से भी बुलाया जाता है। सो व्हाट आर दे? They are the hexose, monophosphate shunt. The second is a pentose phosphate pathway which is known as PPP, और उसे भी बोलते हैं phosphogluconate pathway. Okay, so these are the three names which are of this pentose phosphate pathway. So first of all, we have to understand that where this process is taking place. In case of animals, it is taking place in cytosol. ये जो animals में present होता है, ये cytosol में present होता है. ये process जो present है, वो cytosol में होता है. लेकिन plants में, in case of plants, ये जो process जो होता है in the plastids. Okay. So the first thing is that we have to understand why it is required, and the second thing, where is the position, or where in which organelles it is present. So this is one of the oxidation pathway. The glucose is being oxidized in this way, and it is taking place in the cytosol in case of animals, and in case of plants it is taking place in the plastids. So this process, this whole process of this pentose phosphate, it is taking place in two phases. The first phase is the oxidative phase. First one is the oxidative phase. The second phase which is present, it is known as the non-oxidative phase. Okay, non-oxidative phase. So at first we will discuss how this oxidative phase is taking place and what are its scenario. Okay, so this is the oxidative phase. First one is our oxidative phase. Oxidative phase. So this reaction of this phase is starting from this glucose 6-phosphate. It is not starting from this glucose. It is starting from glucose 6-phosphate. Okay. This glucose 6-phosphate, it is converted into another molecule which is known as 6-phosphoglucolactone. And for this process to take place, in this case, there is requirement of enzyme. So the enzyme which is required is the glucose 6-phosphate dehydrogenase. Glucose 6-phosphate dehydrogenase is the enzyme which is involved in this case, and for its stability it also requires Mg+2. Okay, so what happens in this reaction? What is the output of this reaction? In this case, the NADP plus is being utilized and it is converted into NADPH and H plus. Okay, so this was the first step. So what is the next step now? In the next step, the 6-phosphoglucolactone which is present, it is being converted into another one molecule which is known as 6-phosphogluconate. So during this time, there is utilization of water. One molecule of water is being utilized and H plus is removed. And the enzymes which is required here, it is the gluconolactone. The enzyme which is required, it is the gluconolactonase. Okay, now this is the second step. Now we will discuss the third step. What happens in case of the third step? In the third step, this 6-phosphogluconate which is present, it is being converted into one of the 5 molecule compounds. So this things which was taking place, it was a 6 molecule. Sorry, it was having the 6 carbon containing compound. This 6-phosphogluconolactone, it is also a 6 carbon containing compound. 6-phosphogluconate which is present, it is also a 6 carbon containing compound. The next phase which will be produced, it is a 5 carbon containing compound which is known as ribulose 5-phosphate. Okay, and during this condition what happens, there is release of one carbon dioxide, and here there is requirement of NADP plus, which is being converted into NADPH, DPH and H plus. Okay, so in this case the 6 molecule compound is being converted into a 6 carbon containing compound is converted into a 5 carbon containing compound, and this is the oxidative phase, and the oxidative phase is the irreversible phase. Okay, let us see here the oxidative phase. You can see here the 6 glucose 6 phosphate which is present, it is being converted into 6 phosphogluconate. Here the intermediate steps are skipped. So during this case there is utilization of NADP plus, and from this NADPH is being removed. Okay, now the 6 phosphogluconate which is present, from this there is removal of one carbon dioxide, and by the utilization of this NADP plus it is being converted into ribulose 5 phosphate, and here there is generation of NADPH. So this ribose 5 phosphate which is present, it will be converted next into ribose 5 phosphate. Next, this ribose 5 phosphate will be utilized in making the nucleotides, coenzymes. It will help in the production of this DNA as well as the RNAs. So this was all about the oxidative phase. Now we will discuss about the non-oxidative phase. So here is a detailed way of this oxidative phase, how this glucose 6 phosphate is first converted into 6 phosphogluconolactone. This 6 phosphogluconolactone which is produced, after that it will produce 6 phosphogluconate. The 6 phosphogluconate will be converted into D-ribose 5 phosphate, then it will be converted into ribose alpha ribose phosphate. Okay, this is the phases of the oxidative pathway. The next phase is a non-oxidative phase. This non-oxidative phase which is present, it is a reversible phase. So in this whole case, what we were seeing that one molecule of glucose 6 phosphate was giving 1 molecules of ribose 5 phosphate. So here we will see. So here what happens, one molecule of glucose 6 phosphate was giving ribose 5 phosphate, ribulose 5 phosphate. Okay, and during this time there is also removal of carbon dioxide. One CO2 molecule is removed. So what will happen if there is utilization of 3 glucose 6 phosphate? So here in this case it will give 3 molecules of ribulose 5 phosphate, and here there will be removal of 3 CO2 molecules. So from where we got this? We got this from oxidative phase. Now comes the non-oxidative phase. What happens in case of non-oxidative phase? There is utilization of only 1 glucose 6 phosphate. So the extra 2 molecules of glucose 6 phosphate, it will be recycled. So we will see how the recycling of this phases is taking place. So here let us see, here there is utilization of 3 molecules of, so here there is utilization of 3 molecules of ribulose 5 phosphate. Okay, so if it is 3 molecules of this 5 carbon containing compound, so how many carbon it will be present? So here there will be 3 into 5, that is 15 carbon is present over here. So from this what we will get, we have to generate 2 molecules of glucose 6 phosphate. Glucose 6 phosphate will be produced. Glucose 6 phosphate will be produced, and it will be of 2 molecules. So how many carbon? 6 into 2 equals to 12 carbon. Okay, plus what we will get? We will get glucose 3 phosphate, that is the 3 carbon containing compound. So let us go to the pathway so that we can understand how this process is taking place. Okay, so here like we can see, there is uses of 3 ribose 5 phosphate. So let me first of all write and explain, then we will come to this part. We will explain, we will read it by this help of this diagram. So what happens, we are using 3 molecules of RUBP. Okay, 3 molecules of RUBP is being utilized. So here there is one RUBP, this is the second RUBP, this is the third RUBP. Okay, So from one RUBP we are synthesizing xylulose 5-phosphate. Okay, and from another one we are synthesizing ribose 5-phosphate. From another one there is synthesis of xylulose 5-phosphate, and these are xylulose 5-phosphate and ribose 5-phosphate, they are the epimers. Okay, so for giving the two 6 carbon containing compound, like what we have seen here, that we have to give 2 molecules of glucose 6 phosphate, because here only one glucose 6 phosphate should be utilized here. So 2 molecules of glucose 6 phosphate need to be removed, or it needs to be recycled. So this recycling is done by this non-oxidative phase. So we will see how this recycling is being done, and how only one glucose 6 phosphate is being carried forward for the whole oxidative process to take place. Okay, so here what happens, there is utilization of this 3 things, 3 RUBP, and from this 3 RUBP 2 molecules of glucose 6 phosphate will be recycled. So here there is involvement of one enzyme. So here first there is utilization of this cellulose 5-phosphate and ribose 5-phosphate. So for this, now there is utilization of one enzyme which is known as transketolase. It is known as transketolase. So what this transketolase will do, it will help in transferring 2 carbons. When this enzyme will work, what it will do, it can help in the transfer of the 2 carbons from one molecule to the other molecules. So here there is 5 carbon containing compound is present. So if this transketolase is working, that means it is having the ability that it will transfer 2 carbons. Okay, so if it is transferring 2 carbons from this cellulose, so how many carbons will remain? It will give 3 carbons here. This 3 carbon which is present, compound which is present, it is known as glyceraldehyde 3-phosphate. If this 2 carbon is being transferred, this 2 carbon from this cellulose will be transferred to ribose 5-phosphate. So now this ribose 5-phosphate which was present, it will be converted into a 7 carbon containing compound, and the 7 carbon containing compound which is present, it is known as pseudoheptulose. Okay, so here now there are glyceraldehyde 3-phosphate which is a 3 carbon containing compound, and a pseudoheptulase which is a 7 carbon containing compound. So now there is involvement of another one enzyme which is known as transaldolase. So here another one enzyme will come and bind which is known as transaldolase. Okay, what this transaldolase will do, it will help in transferring the carbons from pseudoheptulose, and it will transfer the carbons from this pseudoheptulose. Okay, so here how many carbons will be transferred? Here 3 carbons will be transferred at a time. Okay, so if 3 carbon is being transferred from the pseudoheptulose, it will be taken up by the glyceraldehyde 3-phosphate. Now this glyceraldehyde 3-phosphate, which was a 3 carbon containing compound, it will be converted into 6 carbon containing compound which is known as fructose 6-phosphate. And the pseudoheptulose, after it has taken 3 carbon, after this enzyme has taken 3 carbons from the pseudoheptulose which was a 7 carbon containing compound, now it will have only 4 carbon containing compound which is known as erythrose 4-phosphate. Okay, it is having erythrose 4-phosphate which is having a 4 carbon. Okay, now this erythrose 4-phosphate which is present, okay, from where it will take next, it is a 4 carbon containing compound, but at last we have to give the 6 carbon containing compound which is known as glucose 6-phosphate. Okay, so here there was fructose 6-phosphate. This fructose 6-phosphate which is present, it will be converted into glucose 6-phosphate which is a 6 carbon containing compound. So one glucose 6-phosphate is here, it is being regenerated. Okay, now from this erythrose phosphate, another glucose 6-phosphate will be regenerated. So here comes the third xylulose. Okay, here not third, it is the second cellulose. Okay, this was the first cellulose and this is the second cellulose. Now this cellulose will work, it will interact with this erythrose 4 phosphate. Okay, and here which enzyme will work? In this case, the enzyme which will work is the transketolase. It is the transketolase. If transketolase is working, that means how many carbon it will transfer? It will transfer 2 carbon. Okay, from 5 carbon, 2 carbon is being given to this erythrose 4 phosphate. Now, so what happens here? That there is 6 carbon containing compound which is known as glucose 6 phosphate. This is the second glucose 6 phosphate which is being given or regenerated. Okay, now the cellulose 5 phosphate which is present, which was having only 5 carbon, and with it, now it will be converted into a 3 carbon containing compound which is known as glyceraldehyde 3 phosphate. Okay, in this way, this 2 glucose 6 phosphate, they are being regenerated, and this glyceraldehyde 3 phosphate which is present, which is a 3 carbon containing compound, it will be utilized in the next step. यहाँ पे आप देख सकते हैं राइबोस 5 फॉस्फेट जो प्रेजेंट है, इसका एपिमर ही जो है इट इज द जाइलुलोज 5 फॉस्फेट. फर्स्ट व्हाट हैपेंस, ये जो एक मॉलिक्यूल्स, तो 3 मॉलिक्यूल्स हम यूज कर रहे थे, राइबोस 5 फॉस्फेट का 3 मॉलिक्यूल्स यूज हुआ था. सो यहाँ पे वन ऑफ द राइबोस 5 फॉस्फेट जो है वो कन्वर्ट हो जाएगा जाइलुलोज 5 फॉस्फेट में, जो कि है इसका एपिमर. सो यहाँ पे एक एंजाइम काम करेगा विच इज नोन एज ट्रांसकीटोलेज. ट्रांसकीटोलेज जो प्रेजेंट है, व्हाट इट विल डू, इट विल हेल्प इन ट्रांसफरिंग 2 कार्बन फ्रॉम वन प्लेस टू द अदर. ओके, सो यहाँ पे जो वो काम करेगा, इट विल ट्रांसफर द 2 कार्बन फ्रॉम जाइलुलोज इनटू राइबोस 5 फॉस्फेट, और ये देगा क्या? स्यूडोहेप्टुलोज 7 फॉस्फेट देगा. ठीक है, नेक्स्ट एंजाइम विच विल वर्क, सॉरी यहाँ पे तो 7 कार्बन कंटेनिंग कंपाउंड हो गया, यहाँ से 2 कार्बन चला गया यहाँ पे राइबोस 5 फॉस्फेट में, सो ये हो गया 7 कार्बन. इसके पास कितना कार्बन प्रेजेंट होगा? नाउ इट विल हैव 3 कार्बन मोर. सो जो 3 कार्बन कंटेनिंग कंपाउंड जो प्रेजेंट है, इट इज अ ग्लिसरल्डिहाइड 3 फॉस्फेट. या इस दोनों के पास, दैट इज अ स्यूडोहेप्टुलोज 7 फॉस्फेट और ग्लिसरल्डिहाइड 3 फॉस्फेट जो प्रेजेंट है, इन दोनों मॉलिक्यूल्स के पास काम करेगा और एक एंजाइम which is known as transeldolase, and it will help in converting into fructose 6 phosphate in and erythrose 4 phosphate. ये जो fructose 6 phosphate जो present है, ये एक 6 carbon containing compound है, and erythrose phosphate which is present, it is a 4 carbon containing compound. ये जो pseudohaptulose 7 phosphate जो present है, वो convert हो गया erythrose 4 phosphate में, और glyceraldehyde 3 phosphate जो present है, वो convert हो गया fructose 6 phosphate में. ठीक है, सो यहाँ पे जो एरिथ्रोस 4 फॉस्फेट जो प्रेजेंट होता है, वो काम किया और एक जाइलुलोज 5 फॉस्फेट के साथ मिलकर, यहाँ पे और एक प्रेजेंट था जाइलुलोज 5 फॉस्फेट. सो हियर एरिथ्रोस फॉस्फेट जो कि 4 कार्बन कंटेनिंग कंपाउंड, वो 5 कार्बन कंटेनिंग कंपाउंड के साथ मिलकर काम किया इन प्रेजेंस ऑफ द एंजाइम ट्रांसकीटोलेज. ट्रांसकीटोलेज क्या करेगा? 2 कार्बन को ट्रांसफर करेगा. ठीक है, ट्रांसकीटोलेज जो प्रेजेंट है वो कन्वर्ट करेगा 2 कार्बन को. सो यहाँ से 4 कार्बन के पास और एक 2 कार्बन आकर मिल गया, तो वो हो गया fructose 6 phosphate, and it is being transferred from xylulose 5 phosphate. Now from 5 carbon 2 is being eliminated, so here remains 3 carbon which is known as glyceraldehyde 3 phosphate. So यहाँ से जो fructose 6 phosphate हुआ था, वो कन्वर्ट हो गया glucose 6 phosphate में, because they are the isomer. और ये जो fructose 6 phosphate जो प्रेजेंट है, वो फिर से इस स्टेप का जो fructose 6 phosphate है, वो भी कन्वर्ट हो गया glucose 6 phosphate में. So here 2 molecules of glucose 6 phosphate is being given, or it is being recycled. Okay, so